welcome back. Don't forget to like, share and subscribe to my channel. Today we will look at the digestive system in the organisation topic with tips and exam questions to end with. We will look at the breakdown of food and digestion. The breakdown of food in digestion is catalysed by enzymes. Enzymes are produced by specialised cells, and these specialised cells are in the glands and the gut lining. Different enzymes will break down different foods. Foods such as carbohydrates, proteins and fats. Okay, let's have a look at an overview of the digestive system and the journey of the food. The food starts in the mouth where it has salivary glands, then it passes through to the esophagus. The food then goes to the stomach. You can also see box 3A, 3B and 3C. The food does not go through these structures. These structures only help with the breakdown of the food, and we will look at this later on in the video. The food from the stomach goes into the small intestines, which is at number 4. From the small intestines it goes into the large intestines, then it goes into the rectum and eventually is released out of the body through the anus. So let's have a look at each box in a bit more detail. The salivary glands are contained in the mouth. The salivary glands release saliva. The saliva contains the enzyme amylase. The amylase breaks down the carbohydrates into simple sugars such as glucose. The food then becomes soft and goes into the esophagus, which is a pipe that connects the mouth to the stomach, and the food is called bolus. The process of peristalsis happens in the esophagus. This is when the muscles contract and relax, and they do this to help push the food down the esophagus. From the esophagus, the food goes into the stomach. The stomach has several functions. It contains something called gastric juices. Gastric juices contain the enzyme proteases, and one of the enzymes is called pepsin. Pepsin breaks down protein into amino acids. The stomach also contains hydrochloric acid. The hydrochloric acid provides the correct pH for pepsin to work in. This pH is pH 2, which is acidic. The hydrochloric acid also kills the bacteria. As well as this, the stomach is involved in churning the food, which makes something called chyme. Before the food can be passed into the small intestines, the condition of the small intestines needs to be correct so the enzymes can work properly. To help do this, the liver produces something called bile. The bile is responsible for providing an alkaline condition for the enzymes to work in. It also emulsifies fats. This means that the fats are bigger in size and it makes them into smaller fragments. If they are smaller, this means that the enzymes can easily attack them and the process of digestion and the breakdown of food can be quicker and more effective. Once bile is made in the liver, it needs to be stored, and this is done in the gallbladder, which is this structure here. As well as this, the pancreas is also involved. The pancreas produces something called pancreatic juices. Pancreatic juices contain enzymes such as amylase, protease and lipase. A tip to remember that inside the pancreas, the amylase is called pancreatic amylase, and inside the saliva, the amylase is called salivary amylase. They both break down carbohydrates into simple sugars. Now that the pancreas has passed its pancreatic juices with all the enzymes into the small intestines, and also the liver has produced the bile and the gallbladder has passed this bile into the small intestine, it now contains the correct condition for the enzymes to work in. So the small intestine contains the amylase, the protease and the lipase, and with this it can complete the digestion because the bile has produced the right pH for these enzymes to work in. Once the digestion is complete, the nutrients are then absorbed into the bloodstream. The small intestines are shown here. Once the nutrients are absorbed in the bloodstream, the large intestines, shown here, are responsible for absorbing excess water from the food. Eventually, the rectum, shown here, stores the feces, which is the indigestible food, before it is removed out of the body through the anus. The tip to remember is that in the stomach the condition is acidic, whereas in the small intestines the condition is alkaline. That means the pH for the stomach is around about 2, and the pH for the small intestines is above 7. This brings us to the last section, exam questions. Question 1. Enzymes are sometimes used in industry. Draw one line for each enzyme to the correct industrial use of that enzyme. Here we have the enzymes and here we have the industrial use. This is worth 3 marks. Let's analyse the statement. So we have starch, which is a carbohydrate, so this would have to be to do with carbohydrates. Removes grease, so grease is fat, so this would have to do with lipases. Pre-digests proteins and some baby foods, this would have to do with proteases. Changes glucose syrup into fructose syrup. This is to do with sugars, so we're slightly unsure which one this will go with, but we can work out this with some exam techniques. Let's have a look at those. So let's match the ones we do know. So carbohydrates is the one that's to do with carbohydrates, which is the starch. Isomerases, well, this is the one that we're a little bit unsure about. Proteases is the one that matches with the proteins. We have 2 options left. We have removed grease, stains from pores, and changes glucose syrup into fructose syrup. So this means that it can't be this one here, because this is to do with fats, it's to do with lipases. The enzyme isn't called lipase, it's called isomerase, so the option must go there. So this is a way how you can tackle some questions that you actually might not be sure about, but you can use the information given that you are sure about to actually find out the answer for the one that you're not sure about. Let's look at question 2. Lipases is an enzyme that digests fat. Complete the equation to show the digestion of fat. Use the correct answer from the selection below. So our selection is glucose, glycerol and glycogen. We are shown fats being broken down by the enzyme lipases, so we know fats can be broken down by lipases to fatty acid and glycerol. Not glucose, because glucose is a sugar and glycogen is a word that we haven't come across yet, so our answer must be glycerol. This is worth 1 mark. Let's have a look at the last part. Name one organ that makes lipases, worth 1 mark. Well, if we think back, the pancreas was an organ that made all 3 types of enzymes. It made lipases, proteases and amylase. Our linking word today is enzyme. So if we think about enzyme, we should think about proteins, we should think about biological catalyst, about digestion. When we think about digestion, we should think about absorption of nutrients. When we think of digestion, we should also think of the 3 different types of enzymes: proteases, carbohydrases, lipases. A protease, we should think of pepsin. When we think of pepsin, we should think of the stomach. We think of carbohydrases, lipases and protease. We should also think of the small intestines. Think of small intestines, we should think of the villi. Think of villi, we should think of large surface area. Think of large surface area, we should think of effective absorption. I'm sure you can think of lots of other linking words that we have studied so far to do with enzymes. Thank you for watching. Please join me on my next video. Don't forget to like, share and subscribe to my channel.